Hello students, in this video we will discuss about transcription in eukaryotes. In the previous video we discussed about transcription in prokaryotes. Now it is the turn of transcription in eukaryotes. Okay, and the mechanism will be same, but only thing is that this transcription in eukaryotes occurs in the nucleus. And transcription, of course, is required for the synthesis of RNA. And already we have come to know that all types of RNAs are formed only through this process called transcription. And we saw that all RNAs, mRNA, tRNA, rRNA, they're all formed under the influence of the enzyme DNA-dependent RNA polymerase enzyme in prokaryotes. But when we come to eukaryotes, in the nucleus of eukaryotes, you will find three types of RNA polymerase enzymes. Very simple. What are those three? RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Okay, that means these enzymes will synthesize different types of RNAs. This indicates that there is division of labor in the eukaryotes. Okay, and you had to memorize what are those RNAs catalyzed by the enzymes. Okay, RNA polymerase 1 will synthesize 5.8 S rRNA, 18 S rRNA, 28 S rRNA. That means what? Ribosomal RNAs. Don't forget that ribosomes are composed of proteins and RNA. Which RNA makes that one? Ribosomal RNA. And this ribosomal RNA is present in bits, and they are measured in Swedberg units. That's why we are using like this: 5.8 S rRNA, 18 S rRNA, and 28 S rRNA. And one is missing here. Okay, that is 5S rRNA. 5S rRNA. But you can see that these three ribosomal RNAs are catalyzed by RNA polymerase 1. And now, when we come to the second one, RNA polymerase 2, so it catalyzes the formation of hnRNA. What is this hnRNA? Heterogeneous nuclear RNA. Heterogeneous nuclear RNA. Because in eukaryotes, mRNA is not directly formed. So instead, what is formed? Heterogeneous nuclear RNA is formed. What is it actually? Means heterogeneous nuclear RNA will have exons and introns. Okay, coding information, non-coding information. And of course, here we should not blame RNA, because it just copying as it is what is there on DNA. The DNA itself is having exonic information and intronic information, and this is called split gene. Okay, that's why even this RNA, when it is formed, it brought both informations, exons and introns. That's why post-transcriptional processing is required for the RNA that is formed in eukaryotes. That's why after transcription, after processing, that hnRNA is converted into mRNA. I hope you have noticed this point. So there is no processing required in the case of prokaryotes. In prokaryotes, mRNA is directly formed. But in eukaryotes, mRNA is not directly formed. So only hnRNA is formed, and hnRNA later is converted into mRNA. However, that we will discuss later. And when we come to the third enzyme, RNA polymerase 3, and this RNA polymerase 3 will catalyze the formation of tRNA, that is transfer RNA, 5S ribosomal RNA, and scRNA. What is this scRNA? Okay, small cytoplasmic RNA. Small cytoplasmic RNA. And what is snRNA? Small nuclear RNA. Okay, small cytoplasmic RNA and small nuclear RNA. So these okay, one, two, three, four types of RNAs are catalyzed by RNA polymerase. But here you have to remember one thing. So don't come to a conclusion that in eukaryotes three types of RNA polymerase enzymes are there. Actually, four types are there. How four types? What is that fourth one? RNA polymerase. What is that RNA polymerase? Where you will find this one? Don't forget that the eukaryotes will have membrane-bound cell organelles. And among these membrane-bound cell organelles, there are certain cell organelles which are bounded by two membranes or double membrane-bound cell organelles. Okay, and what are those double membrane-bound cell organelles? Chloroplast and mitochondria. And this chloroplast and mitochondria will have this genetic material in the form of DNA. A circular naked DNA is present in them. So in the stroma of chloroplast and in the matrix of mitochondria, you will find this double-stranded circular naked DNA. So, and whenever DNA is there, that means the DNA will perform its usual functions like replication and transcription. But in that chloroplast and the mitochondria, transcription is catalyzed by one enzyme, that is RNA polymerase enzyme. That's why these chloroplasts and mitochondria are considered as what? Prokaryotes in eukaryotic cells. Okay, just consider it like that. They are cell organelles, of course, but they resemble the prokaryotes in the presence of these circular naked double-stranded DNA and same 70S ribosomes. Okay, so that's what you have to remember. So now I hope you got this point. That now can you tell me that how many types of RNA polymerase enzymes will be present in the eukaryotic cells? Okay, four types. What are those four types? RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, and RNA polymerase present in chloroplast and mitochondria. Okay. Now, when we come to this mechanism of this transcription in eukaryotes, it happens just like in prokaryotes. So here also we will find the three steps. One is initiation, second one is elongation, and the third one is termination. And let us, for example sake, let us take this RNA polymerase 2, okay, and it is catalyzing this reaction. So this is RNA polymerase 2 here. And already in the previous video we discussed that this RNA polymerase is consists of polypeptides, okay, alpha 2, beta, beta dash, and omega. And we call that protein part as what? Apoenzyme, and it has a cofactor, and both of them together make what? Holoenzyme. So here also you will find what? Sigma factor, and that sigma factor starts this transcription. And where? That means you know that this is a transcriptional unit, and in the transcription unit we find three zones or three regions: promoter site, promoter region, structural gene, and terminator. Okay, and this promoter region is identified only by sigma factor. That's why this holo enzyme occupies this one and now starts transcription. And of course, already we know that transcription occurs only on one strand of DNA, that is called template strand. Okay, and now it is bringing triphosphoric nucleosides according to the complementarity, which we discussed already. So now it started, and now as soon as this polymerase moves into the structural gene area, the sigma factor will come out, because its function is only to recognize the promoter site. Now, this enzyme, what enzyme it is? Okay, that's correct, RNA polymerase 2. But which one is it? Okay, core enzyme or holo enzyme? Core enzyme. So that one will continue, and as a result of which this RNA, okay, is elongating. This is called elongation step. And by the time it reaches this one terminator, and the terminator for recognizing terminator site, one factor is required, and that is what rho factor. Okay, now you can see that this enzyme is associated with the rho factor, and as a result of which, so this RNA, nascent RNA which is formed, will be released. And once that nascent RNA is released into the nucleoplasm, okay, I hope you're understanding, not in the cytoplasm, into the nucleoplasm, immediately this enzyme along with rho factor will come out. And already we discussed that during this process, and how the hydrogen bonds will break. So hydrogen bonds are broken by this RNA polymerase enzyme itself. There is no involvement of helicase enzyme during transcription. So that is what the same process, and only thing is that the RNA which is formed here, you can't call it as mRNA. This is called hnRNA. This is called hnRNA, that is heterogeneous nuclear RNA. Heterogeneous nuclear RNA. Okay, that's why you see this heterogeneous nuclear RNA cannot participate in translation. Okay, that's why before it comes out into the cytoplasm, it should be processed, and that is called post-transcriptional processing. And we will discuss about that post-transcriptional processing now. Now, let us say this post-transcriptional processing. Okay, and it is nothing but what conversion of heterogeneous nuclear RNA into messenger RNA. That is what post-transcriptional processing. So while this transcription is occurring, this capping will happen at the same time. As soon as remember, this hnRNA is going on forming. So okay, this is what end? 5' end. Okay, and this is 3' end of RNA. So while this is formed, so a unusual, okay, guanosine is added. That is methylated guanosine triphosphate is added. So which can be written like this: methylated. So I'm writing M here. PPP. That is methylated triphosphate is added. At what end? 5' end. Okay. So this attachment of methylated guanosine triphosphate methylated guanosine triphosphate to 5 prime end of HNRNA is called what? Capping. And this capping is required for identification of the ribosome. So ribosome, it has to identify the mRNA as soon as it enters into the cytoplasm. And only when it binds to this mRNA, then only translation process will occur. So that's why this capping is required for that purpose. Okay, and next, as soon as transcription is completed, at 3 prime end, polyadenine chain is added. Polyadenine chain or poly A chain. Poly A chain. A chain of many adenines is added. That is called tailing. And now let us say this is the most important one. Okay, this is 5 prime end and this is 3 prime end, and here there is guanine, methylated guanine triphosphate, and here there is adenine polyetail. So this is what we got, and we call it as what? HNRNA. Now this HNRNA is having okay, uh, exons and introns alternately. Exons, introns, exons, introns, like that. And know that exons are the coded information or useful information, whereas intron is useless information. And now one enzyme will come, and that enzyme is what? Okay, endonuclease. And it will break these exons and introns. Break exons and introns. And now, you see here some snarps will come here. They are attached like this to intronic regions. Okay, and what are these snarps? Small nuclear ribosomal proteins. Small nuclear ribosomal proteins. Small nuclear ribosomal proteins. And remember that whenever these proteins come, and you see, I have to show you once again. So they push this stand like this. Okay, push. Okay. So now you will find the introns are pushed. Okay, and now these introns appear like loops along with what these snaps, and these are called spliceosomes. Spliceosomes. And now you can see that what has happened. So the introns are separated from the exons. And now one enzyme will come, and that is the ligase enzyme. And this ligase enzyme will join all these exons, as a result of which, okay, now This strand is composed of only exonic information, that is coded information. So this is called mRNA. mRNA, messenger RNA. Okay, so this step is called what? Splicing. But I hope you are understanding this procedure or the process. So first, at the end of a transcription in eukaryotes, what is formed? Not mRNA, hnRNA, that is heterogeneous nuclear RNA, and this heterogeneous nuclear RNA, nuclear RNA, is now is processed into what? Messenger RNA. But this step is absent in prokaryotes. So, and remember once in NEET examination only they asked this question: in which organisms spliceosomes are not formed? Very simple question actually. And what is the answer? In prokaryotes. Why there is no splicing there? Why no splicing? Because mRNA is directly formed in prokaryotes. Okay, that is about transcription in eukaryotes.